Howdy everybody, you're listening to Blue Jeans and Boots, where genetics grad students talk about how to navigate a PhD program while trying to stay sane. We also invite faculty from our interdisciplinary program here at Texas A&M onto the show and pick their brains about their research, what it's like being a principal investigator, and their journey to becoming a professor here at A&M. In this episode, we will be talking to Dr. Matt Sachs. Dr. Sachs is a professor in the Department of Biology at Texas A&M University. He received his Bachelor of Arts in Biochemistry from Cornell University and a PhD in Biology from Massachusetts Institute of Technology. Dr. Sachs' research focuses on the mechanisms of controlling protein synthesis, such as in Neurospora crassa. My name is Charles Mitchell, and I'll be your host today. So, howdy, Dr. Sachs. Would you like to give us a brief info on your bio and how you got into your research studies?
Thank you, Charles. Yes, as Charles said, my name's Matt Sachs. I'm a researcher, and my brief bio is I started out at Cornell, and I thought I was going to be a practicing clinical doctor. And in the late 1970s, Tom Maniatis and many others, some others, but Tom Maniatis in particular published this very beautiful work on cloning and analysis of globin genes that gave me the idea that life could be better served for medical reasons, for pursuing a medical career of doing genetics, molecular genetics. And from there I went to MIT and I worked in a laboratory, Tom Rajmandari's laboratory. Tom and Gobind Khurana came together to MIT from Wisconsin. Gobind had won the Nobel Prize for discovering the genetic code, and this got me very interested in protein synthesis. And from there, I went to Stanford, Charlie Yanofsky's lab, who, as some of you in the audience may know, was a terrific scientist. And among the genetic mechanisms that he discovered for how cell regulation occurs is a mechanism by which a leader peptide synthesis in an mRNA controls protein synthesis, controls mRNA translation. And not surprisingly, with that, I became quite interested in making a career of understanding protein synthesis, and that has been largely what brought me here.
Cool. So you mentioned medical school, so a lot of grad students, including myself, were pre-med at one time. So can you talk a little bit more about what drove you into science and what your interests in science were beyond what you've already stated?
Yes. I think that my interest in research science is captivated by the idea that, use a metaphor, nature's book is magnificent, and the ability that we have now and continue to gain, and that goes from the 1970s when I started to the 2020s, that our ability to read this book just gets to be greater and greater. And it's wonderful to see the blueprints and the mechanisms by which those blueprints are brought into reality in living cells. So I think there is a much more of a, call it an aesthetician or an aesthetic view of science, as opposed to I'm going to cure X or I'm going to cure Y view of science.
So you have a very established CV and resume, and you've been lots of places. So what was the one thing or things that brought you here to Texas A&M specifically? Like, what was the draw? What was the passion here?
Thank you, Charles. This is a question that'll have to be a little bit technical. Okay. I was on the faculty at Oregon Health and Science University essentially for 17 years. My institution where I started in Oregon merged with them, and earlier on I had been joint faculty. And I was working on protein synthesis, and as Charles mentioned, I studied Neurospora crassa really since about 1980. Deb Bell-Peterson, a terrific faculty member in the Department of Biology and also in the genetics program, essentially knew that biology at Texas A&M was expanding and asked me to apply for the position. And she also was interested in the role of protein synthesis in the cell's circadian response, so that there's a day and night cycle in protein synthesis in cells. And our labs are actually adjacent to each other here. So being with Dr. Bell-Peterson was a very big draw, but also importantly, I was already working with scientist here who actually retired shortly after I came here, but had worked out mechanisms, or methods I should say, for putting what are called photoaffinity cross-linkable analogs into nascent peptides. And so we were already collaborating to put these small nascent peptides in, which are encoded or by RNA and are being translated by the ribosome, so that I could see how the nascent regulatory peptides I was interested in studying changed their conformation inside the ribosome. So Oregon to Texas versus just across essentially Wellborn Avenue in Texas was both terrific ideas to move. More generally, I really like being at a Tier 1 research institution compared to a medical school. And for me, all things are local. I mean, Oregon was a very beautiful place to live, and I had great colleagues at the medical school. But I really wanted to be back at a large Tier 1 research institution. So all of these were good reasons for me to come career-wise. And I just want to briefly add, and the people here are also fantastic, not just at work, but in real life as well.
Yes, I can totally agree that those were some of the same reasons that I was personally drawn to Texas A&M. And so you talked about how important collaborations and connections are in science. So you've previously collaborated with the Broad Institute and other researchers on functional genomics projects. What has been one of the most surprising discoveries that have arisen from these collaborations? And if you want to give some more details about those.
Yes, well, surprising discoveries that came from the Neurospora genome. This is a small surprise. This isn't a radical surprise for the planet. But for me, one of the great scientists in Neurospora, Eric Selker, had shown that there are very few repeated DNA sequences of any length at all in Neurospora. Yet there were some families of genes where there were actually proteins that were, let's say 95, I'm making up a number at some level, but 95% identical at the amino acid sequence level, and yet they were not being evolutionarily driven out of the Neurospora genome. It turned out when we looked at these genes that encoded these proteins more carefully, they had different intron arrangements that essentially broke up the homology blocks to a level where they would no longer be subject to the genome defense mechanisms that inactivated gene sequences that were highly similar. So as I say, that's a small one, but that was the kind of information that you could only get from these kinds of large-scale analyses.
So what are some of the real-world applications that your research has that might surprise some people? So as someone who does not work with Neurospora crassa, what are some details that you can share with people outside of your field to get them interested in what you do?
Yes, that's an excellent question. A little bit of background, that the fungi are humans' closest relative, that although at one point fungi were in departments of botany because they were thought to be plants without chlorophyll. Once people began to look at the genomes of organisms, they saw that fungi were indeed not plants without chlorophyll, but much more closely related to animals than plants. Many of the best work on model organisms has been done in the Saccharomyces cerevisiae, the budding yeast. And Saccharomyces is an incredibly great organism to work with, but it differs in many respects from most other fungi. It has a streamlined lifestyle, and Neurospora in some respects resembles humans more. But doing comparisons to metazoans, animals, and to other fungi gives opportunities for greater understanding of similarities and differences. With respect to my personal work, I am more a fungal biologist, or a scientist who's a biochemist and a geneticist who works on fungi, than I am a mycologist, somebody who tries, or who does get a greater sense of how fungi fit into the larger system of life on Earth. And what we've learned about Neurospora protein synthesis has provided, I think, real contributions. Their significance can be assessed, but there were certainly novel contributions to understanding how nascent peptides stall ribosomes during protein synthesis, and in that way control the regulate the expression of downstream genes. And some of the upstream open reading frames that encode these peptides are highly evolutionarily conserved, but at least the one we're studying now is absent from Saccharomyces, what are called Saccharomyces sensu stricto, but present across all of the other fungal lineages. The Neurospora work is part of a larger tapestry. But if I may just switch gears a little bit, we wanted to get at the structure of a nascent peptide inside the ribosome, and there were no existing structural studies of Neurospora ribosomes or ribosomes from any of the fungi outside of Saccharomyces. And we decided to look first at ribosomes that are arrested in translation, not by a nascent peptide, but by a drug. Drugs that stall ribosomes are considered, in at least one of them, is considered to be an anti-cancer agent. When we did this work, we made a contribution to the structure of the ribosome that was stalled by cyclohexamide with nascent peptides still in the ribosome tunnel. We could visualize what's called the peptidyltransferase center, which is the site of protein synthesis, where you get the carboxyl group and the amino group essentially combining to make an amide bond. And we saw quite clearly that, and this is a little technical, we saw quite clearly that the ribosomes were blocked in a pre-translocation state where the nascent peptide had transferred to the A site tRNA. And nobody had seen this previously. One person in one group, Scott Blanchard's group, had seen this from single molecule analyses of where tRNAs and peptide was in the ribosome. But this also has turned into a contribution. And another aspect of that, and I'm going to go now a little more wide here, that there are some fungi that are naturally cyclohexamide resistant. And among these fungi are species of Candida, such as Candida albicans, which is, some of you may know, a major fungal pathogen of humans. unimmunocompromised, non-immunocompromised people get Candida infections, but if you are immunocompromised, it's quite serious. They're naturally cyclohexamide resistant. As a story, we worked with a group, several groups, and were able to show the basis of the cyclohexamide resistance was a change in the structure of the ribosome at the cyclohexamide binding site. Why am I going on about this? It turns out that there are groups of fungi that are naturally cyclohexamide resistant, and cyclohexamide is made by an actinomycete bacterium, and many of the fungi that are naturally cyclohexamide resistant can live in the ground or in beetle nests, or that's not beetle burrows, I'm not getting that technical term right, along with the bacteria that produce cyclohexamide. So there's an example where a natural drug resistance is occurring in an evolutionary survival fight. So as I say, nature, the whole tapestry of nature, I find to be fascinating.
Yes, that's all very interesting. So you talked about a few technologies or techniques that you used in your lab, but which ones are you most excited about using now and in the future? And if funding and resources were unlimited, what's your dream experiment that you would love to conduct?
That's a great question. What is the dream experiment? In dream approaches versus dream experiment. Well, first, I'm teaching microbiology, and we talk about Alan Hershey, who did the famous experiment which showed that DNA went into bacteria and the protein stayed outside, which confirmed earlier very elegant work by Avery that DNA was the genetic material. And to do this, Hershey basically did short pulses of bacteriophage virus infection and then used a blender to disrupt the phage. But he has a saying, and I've never actually read the direct quote, but people talk about what he called as Hershey heaven, which was doing the same experiment over and over, and each time getting a great result, and each time having it be as if it were the first time. Now, that is many of our view of what Hershey heaven, what science is, and is happiness in science. Now, I don't necessarily want to do the same experiment over and over, but for me, I would like to know how nascent peptides act kind of like fish bones in the proverbial throat of the nascent peptide tunnel, the throat of the ribosome, to stop protein synthesis. And to do that, we ultimately will rely on structure to get at that, although there's a lot of biophysical experiments we can do and biochemical experiments that also will lead to that. That's a very specific question, but that's the current focus of the NIH funding I have.
So as a current grad student, as you know, science requires a lot of perseverance. So are there or have there been any moments in your research or career where you almost gave up on a project, and then, or how did you push through that? And what's your advice to other people currently going through those struggles?
Yes, the mojo question.
Yeah.
I'm not going to answer the mojo question directly, but I do want to mention very much what was a Zen rap on the head when I was a graduate student, when I was cloning genes for the nuclear encoded subunits of cytochrome oxidase. That was my thesis project. And I was doing a very complicated procedure where I was using methylmercury hydroxide gels to purify RNA. Then I was translating those RNAs, those fractions of RNA in cell-free systems, and then immunoprecipitating antibody against cytochrome oxidase to get the fractions of RNA that were enriched. And then I was doing, and this is real old school, DG-DC tailing of PBR322, and then putting in, or DG tailing, and then putting in tailed cDNAs. And Rick Young at the time had developed what was called the Lambda GT11 system, where you could clone cDNAs directly into lambda phage and then have them express the protein products of the inserts, the cDNA, which is this DNA copy of the mRNA, essentially on the phage or in the phage. In the plaques, you could see that directly and just screen for the inserts with antibody. Dr. Barbara Meyer was at MIT at the time. She had just joined, and I was telling her about my project, and I told her the complexity of it, and she said, Rick Young has just developed this technique. Why don't you use that? And I said, well, I am going to do it this way because this is what I'm doing. And she said to me something I will never forget, and this is why I'm talking about it here. She said, there's a difference between being persistent and being obstinate, and you're being obstinate. So this is a very important thing, that sometimes we have to be willing to see what is possibly a better way and be able to let go. And if I may lecture, as I'm obviously lecturing now, a little bit about this. And I mentioned Hershey Heaven, doing the same experiment, doing the same thing over and over, that it is truly important to understand that we should do the experiments that are best to address the question and be fearless about that.
That's all very interesting. So now we're going to rotate kind of towards grad students and related questions. So as a mentor, what is your preferred mentoring style? And then what skills and qualities do you look for in your grad students to better foster their professional development?
Yes. So one thing about science to me is that although there's a hierarchical nature of position titles, that in the end we all are looking at the same processes, and we can all make insights into those processes. The first thing I feel is critical for being a scientist, and possibly a graduate student being a developing scientist, although we're all developing and hopefully not ossifying, is that we understand that rigor is most critical. So I often say to students, and I'll put it out here, who are your two best friends in science? And I'll get all kinds of answers, but the answer to me is the positive and negative control, right? That we have to steer by our own lights as scientists, that the professor is sometimes not correct or doesn't have the best insights. They may, but independent of that, we need the positive and negative control. If we can do a reconstruction for the positive, we certainly need the negative. So for me, the most difficult time I have with students is when they don't conceptualize that everything's an experiment and nothing's a prep, even when they're doing a prep. I'm not singling out any students here. If you do a transformation of bacteria with insert, like in the old days when you're cloning, and you don't include the positive control of uncut vector and know what the transformation efficiency is and no colonies come up on the plates, we should do that once and never again, because, well, last week it worked is not the answer. So rigor is, I think, not as a mentor. I mean, I value that. That's the thing I privilege most highly. Now, the aspects of working. I mean, there is an old saw that scientists can work. At least in my generation, scientists can work whenever they want as long as it's all the time. Now, we know that was 40 years ago, and people have different views of that, but the key thing for me is wanting to do the work. When students when a scientist doesn't want to do the work and it's a job, then that's problematic. And it's not problematic for somebody in some other situation, but I respond very positively to enthusiasm. And it's difficult for me if somebody is not enthusiastic to mentor them.
I think that goes along with the adage that if you love what you're doing, you never work a day in your life. So that makes sense from a lot of different standpoints. So how do you help students specifically with not only their wet lab bench skills, but also their scientific communication skills, going along with writing and presenting their work?
Yes, I'm going to take this back to undergraduate level. Not long after I came here, the Department of Biology developed a capstone research program for undergraduates, and I was put in charge of it for one reason, because I was new and and I had ideas. And I've, for many years now, taught a capstone, now it's honors capstone, the final semester, where students have to present their work and also write a report that they need to have approved by me to get honors credit, to biology honors credit. There's multiple different honors programs at Texas A&M. I think this is an example of learning by doing and having the experience of going through, for instance, with the initial writing, which is writing, let's say, a literature review of 3 papers, and then that will grow into the full report, of getting a marked up draft with the sea of red often, and understanding that this is part of the process. But you can't in a classroom convey that. You have to go through the experience. And similarly with giving a talk, you have to go through drafts. So I'm certainly not unique in this, but many faculty want their students to submit drafts. And I'll tell a story about this, which isn't me. There's a classic story of a professor who had a student submitted a draft to her, and she gave it back the next day with no comments on it and said, needs more work. 3 weeks later, another draft came and she gave it back, no comments, needs more work. 3 weeks later, another draft came and she said, okay, now I'll read it. And no, that's not me. But there is this element that the writing process is what helps us think about what we're doing. Now, how AI fits into this, I'm not entirely sure at this point. AI is useful, but I've done at least limited studies of having AI summarize a paper that I've read. And yes, the main points are there, but it is no substitute for actually reading the paper. So we're now in this new area of how does this work? And I'm just telling a joke here. In the old days, because I grew up before the internet, photocopying the paper was sometimes the equivalent of reading it, right? And now perhaps getting an AI summary is the equivalent of reading it, but there is no substitute for reading it. And so going back to this idea that, how do we train people? You have to see how things are done. And not that, I'm an emulator, I suppose. I like to see how things are done. I'm not so individually creative that I can just go make a new way of writing a paper or something like that. So I think that for me, that's where it is, trying to get students or be with students as they read and write and present is the key. And also, of course, we know, and I'm going to go a little bit sideways, and we talk about training students, and this is a very difficult area where many of us were used to being criticized, at least having our work criticized. And it's been a very difficult thing, and I've been doing this now for decades, that some people take criticism of their work as criticism of them. And it's so difficult to figure out a way to criticize somebody's work without it being a it's not about you, right? I wouldn't even be criticizing this if I didn't think that you were great, kind of thing. And that's a complicated area that some faculty and some people, not just faculty, don't fully understand. And some are criticizing that person too, so that can be problematic.
No, I think that's a very good piece of advice, because my PI also had the same advice when we got paper revisions back. I took it a little too personally. He goes, "Oh, they're expanding your scientific expertise. They're not attacking you." So I think that's a very good point to tell other grad students and other people working in this profession. So beyond that, going back to your grad school days, can you describe your experience and how you would better advise yourself today if today's Dr. Sachs was going back to that Dr. Sachs and being, "Oh, these are some tips that I would give you"?
Well, I could advise the Dr. Sachs of 3 months ago almost the same way. For me, I wish, if that's the right way to say it, that I kept better notes of certain procedures, which I had at the time, but I did not take with me. That just the ability to go back, and we talk about notebooks often, to be able to go back to a notebook, and of course I didn't bring my notebooks with me, so I couldn't access them, but I wished subsequently that I had better ideas of what it was I was doing on a very pragmatic protocol basis. Now we have many more options for being able to keep track of these things. And so that's what I feel also about the me of 3 months ago. And I faced this, and in fact I brought it up with I teach a seminar course with about 6 students on eukaryotic microbiology. And we read something, we think we'll remember it, and we will remember it for several weeks, but let's say 2 months later, it's like, I remember seeing that paper, right? And I'm still looking for that way. If anybody in the audience has real guidance for me, yes, make a Notion database. Well, it's not that simple. It sounds simple, but it's not. So that's it. I have a fairly good memory, but it's a memory that comes from prompting. I don't have that memory that can pull something out of thin air, but if you give me a little bit of a piece, then I'll remember it. And so that doesn't work for remembering that paper from 3 months ago.
No, I'm very much the same way. I need to do better on organizing some of my stuff. So how would you say you balance guiding students while also allowing them the freedom to explore their own scientific ideas? So when you get grad students, are you leading the charge, or do you guide them and then eventually let them take charge?
Okay, so that's a great question. I was really fortunate when I was in Oregon. I had 2 students to start with. This is 1990. One student went off into ultimately away from biological science. My other student is now a distinguished professor at a major research university. And it was very easy because he had the phenotype of being able to figure things out and work on his own right from the get-go. But I've had certainly many students since then, and I think that it is truly a collaboration where you let people do stuff and figure it out, and hopefully figure out when things go right and when things go wrong, what was it that made it right and what was it that made it wrong. We both know that knowing something in principle is different than having something go wrong after you've invested a lot of time in it and then getting, oh, I knew I shouldn't have done that, is different than I did that and I paid. So you have to allow students that room, right? I mean, any scientist, not just students. I'm using students in a generic way here. We all make mistakes, right? And in some ways, trimming the repeats of the mistakes is where the action is. And doing some kind of inductive logic. So seeing that I'm about to make that same mistake in a different way, yes. Right? That's the trick. And so helping people, and that means also helping oneself to see that is, I think, a critical part of the process. I've had very few students who actively wanted to do things just by themselves. But I've had some who do go out and try new things and then report to me. And that's always very nice, you know. Yeah. Matt, we hadn't thought about doing this, but I just did it, and look. Yeah. Is always, I mean, there is very few faculty, and I think very few people that we will work with ever who hopefully, I mean, there might be some that wouldn't. Hopefully everybody we work with wants us to be creative and try new things.
We've talked a lot about the science, but scientists are not only their science, right? We have lives outside of work, hopefully. So what activities do you like to do outside of science to have fun and to stay sane when you're in a stressful situation?
I've had a passion for many decades, even before biology, for photography. And there was a period of time when I was here at Texas A&M, I worked as a photographer for Texas and did a lot of sports photography. And I enjoy that, but I don't have the cycles to have kept doing that. Well, let me say, though, if you go to the Genetics Society of America site, and you find the fungal genetics conferences for several decades, I guess it is now, I photographed the conferences, and that's turned out to be a remarkable resource for the community to have all of the candid photos of the scientists at the conferences. So I still do that, and that's a not-for-profit, although I could have easily been a for-profit photographer. I am a practicing yogi. I've taught here in town since 2012, and I taught in Portland, Oregon. That doesn't make me a master by any means, but I enjoy I'll call it the rigor of knowing when you're teaching, you actually have to have a plan, and it maintains a certain rigor in the practice. And otherwise, cardiovascular stuff, I've always been a runner, but over the years it's now transitioned to more bicycling, so long-distance bicycling has become something I've done now about 6 years. Got long distance is all relative, but we've got a 70-mile ride this weekend, and the following weekend we'll have 2 70-mile rides. This is my wife and I'm talking about. We do these organized group rides, which also provides a social framework because we know the other cyclists. It's their organized rides. That works out very well.
We heard that you are a hot yoga instructor, or do you do regular yoga too? And then you talked about some of the parallels between teaching yoga and also teaching science, but we were wondering, do you have any favorite poses or challenging poses for yoga that other grad students can get into if they've never done it before?
Okay, that's another great question. So hot yoga, I did teach. The studio where I teach, because of the class schedule, it's a multi-use studio, cannot heat the room and then cycle it back down to room temperature so easily. We're in the 80 degrees F yoga, which is not hot. Hot yoga is typically around 105 F. So I think hot yoga has advantages, but also many people struggle with the heat. So I'm content with the lukewarm yoga. Okay. With respect to poses, there are many limbs of yoga. And Hatha yoga is, of course, what we think of when we think about a traditional yoga class. There are poses that work for some people and not for others. But I think, to me, the critical part of yoga, which is also part of science or any kind of a discipline, is perception. So, launching into, and this is a technical word for yoga, a rapid vinyasa flow, right? And simply flowing so quickly that one never actually feels any of the poses may look like yoga, but without the perception, it's an athletic event, no doubt, but it may not be yoga itself. I think a lot of what I strive for in yoga is the sense of knowing the kind of the boundary of potential energy versus kinetic energy, and being able to adapt through poses, whatever they may be, feeling of the wealth of potential energy that's within us, and a kind of a clear-sightedness that we can use that potential energy to convert it to kinetic energy for the things we love, the people we love, the work we love, and to be a positive force with it. That's the ideal pose. It does not necessarily trikhasana or shavasana, but that's, to me, the reason to practice yoga.
No, I love that. That's very, very cool and very interesting. So we've talked a lot about everything across your whole experience and career. Is there anything we haven't covered yet that you feel is important for graduate students to know today?
Yes. If you had told me when I was a graduate student that I would be a full professor at Texas A&M and that would be my career, I would have said, get out of here. And I say that because we don't really know what the future brings. And so going back to that yoga element, be in the present, do the best we are capable of for our own self, and the future will take care of itself, and I hope for everyone, in positive ways, because there's always going to be adversity, and sometimes that adversity is just pure adversity. The life isn't fair sometimes, there's no doubt, but for most of us, thankfully, life is reasonably fair, and that we just, have some kind of feeling that we can't make the future happen without doing the best we can do for ourselves in the present.
As we wrap up, I want to ask you one final question is, how do you define success, both personally and professionally? And then how would you define science as a career? You can be as broad or as specific as you want to be for this one.
This is a great question, and my first department head, Michael Gold at Oregon Graduate Center, before we merged with OHSU, came in one Sunday, and I was working in the lab, and he put his hand on my shoulder. Literally, in those days, you could do that, I suppose, and said, "Matt, what do you want on your gravestone? That you paid $50,000 in page charges?" Because at that time, page charges were a lot less than now, but that would have been a lot of page charges. And that was my department head, right? So what is success, right? Is that the measure of success professionally? Is it recognition? In some ways, we all want recognition. I'm not telling you want recognition. Many of us want recognition. But where is that recognition, right? I mean, there's people who choose private lives and are spectacularly successful in their private life. Raising a family, if that's what speaks to them, being a very effective clinical physician and not publishing papers, that's success. If it's what we personally want for ourselves, to me, that's the metric, right? The world, there's always, going to be something else. I mean, one of my favorite writers, singers, poets, however we say, Bob Dylan in one of his albums said, you got to serve somebody. We all serve. I mean, there's always something we have to do, and it may be an arbiter of our success whether or not we're serving that correctly, but that's not a real measure of success. I think the success is that we feel that we are doing what is best for us under the circumstances that constrain what we can do.
Today's episode was hosted by Charles Mitchell and produced by Sarah Ingram. This podcast is brought to you by the Texas A&M Interdisciplinary Graduate Program in Genetics and Genomics. If you have any questions about our program here at A&M, please feel free to email us at bluejeansandboots@gmail.com. You can also find us on Facebook, Instagram, and X at Genetics Podcast. Thank you, Dr. Sachs, for coming, and thank you all for listening. We hope you tune in next time.